about transcription. Okay, transcription, the process, it happens inside the nucleus. And when we say transcription in one line, if I say transcription is the process in which DNA, from DNA, RNA, which RNA, messenger RNA is synthesized or prepared. That process in which from DNA, mRNA, messenger RNA is made is known as transcription. It takes place inside the nucleus in eukaryotes, while in prokaryotes it takes place since there is no definite nuclear membrane. It happens on the in the nucleoid. Okay, and then the difference between the prokaryotic transcription and eukaryotic transcription is there are two types of chain, that is polycistronic and we have even done the structure also, no? The transcriptional unit. A transcriptional unit comprises of three things. Number one, transcriptional unit comprises of three things. Number one is the initiator or the initiation point. That means from where the transcription will start on the DNA chain, where the transcription will start, that information, that code where it is present, there the this polymerase enzyme will come and arrange itself in that position where it is this initiator, promoter region of the transcriptional unit. From then come the next point. There is after in the structural gene is a structural gene in the transcriptional unit. The second point, the second part of the transcriptional unit is structural gene. In the structural gene, actually it codes for structural gene actually codes for the type of protein it has to produce. The messenger RNA is the one which carries the information for the particular type of protein. So which type of protein it has to be formed, that information is actually carried in the structural gene. Okay, now the third point is the third thing is the after this is over, it has to be removed. Okay, so it is a terminator area. So in the structural unit, the third A part is terminator. The terminator carries the code, actually code for terminating this event, terminating the process. So that's a code again which will terminate the process of transcription. And then further we learned here was in transcriptional unit of prokaryote is actually there are two types. Okay, polycistronic and mono. The structural gene we are talking about, the second thing, the structural gene in the transcriptional unit, the second part. Okay, comprises of two things, two types of structural gene it has. It can be either monocistronic or it can carry the information of polycistronic. When we say monocistronic, mono means one. Okay, one. And eukaryotes have got monocistronic type of structural gene. Okay, structural gene is in eukaryote is monocistronic. That means the structural gene carries the information at a time only for one type of protein. Okay, so it will later on translate into a new protein, only one type of protein. Okay, every time it can translate it into one protein only. The information carries in the structural gene is only at a time for only one type of protein, that is monocistronic. While in prokaryotes, the structural gene carries the information for more than one protein, for the translation of more than one protein, and that type of protein structural gene is known as polycistronic. Poly means many. Okay, so polycistronic structural gene which comprises of which carries the information at a time for many proteins to be translated, that is known as that is known as polycistronic type of structural gene. That is, this was also over. I'm just giving you feedback, okay? And then we have learned about the difference again in eukaryote. In eukaryote, this process is little elaborate, since it has got two three steps added in it. Number one, the introns which in the structural gene only itself. Again, the structural gene only, there are some uh, there are some informations which has to be cut and removed. Okay, so this informations which are extra, which is not needed for the proper protein to be synthesized, that is cut and removed. So that process, splicing, cutting, and then adding the fragments which are the exons, no, the exons in introns are removed and exons are later on brought together and formed one messenger RNA. This is actually known as an extra part of, I mean, something which is not found in prokaryotes. So this is splicing and what is it, addition of the tailing is there, splicing is there, capping is there. These things you will find in eukaryotes. Okay, these three steps are extra: capping, splicing, and tailing. These three processes are the extra which we find only in eukaryotes. Okay, and this about splicing only we have learned till now. About capping and tailing we'll be learning ahead. Okay, so splicing is nothing but removing the introns which carries, I mean, which we don't need for the protein synthesis for that particular protein. So cutting that and removing and bringing the exons together, that is splicing. Okay, now we will move ahead. Okay, 1 minute.
Messenger RNA or mRNA, transfer RNA or tRNA, and ribosomal RNA or rRNA. The
Three types of
mRNA carries the
RNA.
coding information to the sites of protein synthesis. It helps to put together amino acids to make protein. The tRNA carries each amino acid to the
At this position you can see all the three types of RNA used. This is the process of translation. Okay, they're just showing you that time also, before also I told you, every time when we see these animations, you are getting a review or the views of transcription, translation together. What you see this here, pen, I don't know where it's gone. Anyways, what you see this, no, this long chain kind of thing, this is mRNA, where it is written, you can see here the name mRNA, while is this is mRNA, this chain. Okay, mRNA, and what you can see this this kind of nail like thing, they are nothing but the codes. Okay, nucleotides, I mean the nitrogenous bases, while in this is ribosome. So inside this is having rRNA. Okay, while this is the adapter molecule, the green color, not this one. This is the uh, nucleotide what they bring. While tRNA, this is adapter molecule, we call it as transfer RNA, or it is also known as tRNA. All the three tRNAs are used here. This is the one first RNA, this is the second RNA, third RNA. Okay, all the three RNAs are used for the process of translation, and this is the process happening in outside the nucleus in the cytoplasm. Okay, but we are going to learn furthermore. What I told you about transcription is the formation of this messenger RNA. Okay, this is the messenger RNA which carries the information for the protein, and this adapter molecule, this ribosomal RNA. Okay, fine.
ribosome according to the coded message in the mRNA, while the rRNA provides a mechanism to decode mRNA into amino acids. Transcription, the first process in protein synthesis, occurs in 3 stages: initiation, chain elongation, and termination. In the first stage, RNA polymerase, along with the initiation factor denoted as sigma, binds to the promoter sequence in DNA and initiates transcription. This helps in the opening of the DNA helix and separates the 2 DNA strands. Now, the second stage of transcription, called elongation, begins. In this stage, the RNA polymerase builds a strand of RNA using one of the DNA strands as a template. RNA polymerase uses nucleoside triphosphates as substrates and polymerizes using the law of complementarity. This RNA chain growth takes place in 5' to 3' direction. After chain elongation commences, the sigma factor dissociates from the RNA polymerase and can be reused. The final stage of transcription is termination, in which the polymerase, along with termination factor represented by Rho, reaches the terminator region, and newly created mRNA fall off along with the enzyme. This marks the end of prokaryotic transcription.
Okay, so the process of transcription, this is the process of transcription. Now here, as you understood, this is when I told you there are 3 parts of transcriptional unit, that is we're talking about the parts of transcriptional unit, means we are talking about the DNA. Okay, now when we say the process has got, when you write this in the exam, you have to write in this pattern only, like the transcription. First you will write about where does it takes place. That means it takes place in, okay, and one plate will act as the host, or as a leading strand is the from 3' to 5' acts like a template stand on which the mRNA is formed. Okay, and what is needed is on the initiation point, or on the promoter site of the transcriptional unit. Here what will come here, the the RNA polymerase enzyme. Our polymerase is an enzyme, RNA polymerase, because RNA chain we are making. Okay, and along with the sigma factor, okay, a factor which is known as initiator. Okay, so this will come along with the it will come and sit on the acid. These kind of things helps in the execution of the process very precisely. So every time to initiate its process, not only it needs the RNA polymerase enzyme, but also the sigma factor, which will initiate the polymerase, and it will become active now. Okay, now this active site will begin from, that means where the initiation part is there. Here it will not there is no code for mRNA, but yes, that codes for many the important point, that is from where its process. So very particular. So that is initiation point, and where the RNA polymerase sits there. As its RNA comes there, what happens? The unwinding of this DNA took place, and then from here it will start moving towards this direction. Okay, and then as you can see here, till it, as I said, a part is, see, this much only, like a bubble has formed, right? It is something you have learned in the replication also, 3 times transcription. And then what happens next is the chain elongation. When we say chain elongation, this process is happening on the structural gene, that is the part which is having the code for the information for the particular protein, that is the structural gene, is the part on the DNA where the chain elongation takes place. And as the code, suppose here the code is for the termination, okay, so there the rho factor moves, and to deactivate, sigma factor moves out, and the rho factor joins the polymerase enzyme, RNA polymerase enzyme. And once the sigma factor rho factor joins it, it will dissociate and the RNA will move out, while the messenger RNA, which you can see here, is prepared and it will move out of the nucleus from the nucleopore. Okay.
The stages of transcription in eukaryotes are similar to that of prokaryotic transcription. However, there are some differences as well. Let's understand them in detail. Unlike prokaryotic transcription, which occurs in cytoplasm transcription, in eukaryotes, transcription occurs in the nucleus. There is only one RNA polymerase in prokaryotic transcription. However, eukaryotic transcription involves at least 3 RNA polymerases.
Children, the difference is, we are learning the difference in the transcription process in prokaryotes and eukaryotes. As we have learned, first difference they are telling you is the place, the site. The site for transcription in prokaryote is the nucleoid, since they don't have a definite nuclear membrane. So in a way, it is happening in a cytoplasm itself, okay? Because they don't have a membrane which separates the cytoplasm from the nucleus. So we can say it is happening in the cytoplasm itself, both the process, transcription and translation. While in eukaryotes, since we have a definite nuclear membrane which make it separate from the cytoplasm, inside the nucleus is the process of transcription happening in eukaryotes. So in eukaryotes where it takes place, okay, in eukaryotes where it takes place inside the nucleus, while in prokaryotes it takes place in the cytoplasm. First difference. The second difference is the presence of this the type of RNA polymerase enzyme. Since I told you just now that what you call it, in the structural gene of the transcriptional unit of a prokaryote is polycistronic. The same gene can do all the process, while for many protein it has got same kind of information carried in the same DNA structural gene, while in eukaryote it is monocistronic. So what happens every time, if it has to transcribe mRNA, if it has to transcribe rRNA, and if it has to transcribe tRNA, it has got different RNA polymerase enzyme in eukaryotes. In eukaryotes, the things are little elaborate or complicated, we can say little. The reason is the organism is little complicated, right? So in prokaryote, only one RNA polymerase enzyme is enough for the transcription of all type of RNAs. Okay, there are 3 types, no? tRNA, mRNA, rRNA. While in while for eukaryotic type of transcription, what happens, for each type of mRNA, different type of RNA polymerase enzymes are there. We're going to learn about that now, that what are the different types of RNA polymerase enzymes and which are used for different types of transcription of M, T and rRNA.
The RNA polymerase 1 transcribes various rRNAs. On the other hand, RNA polymerase II transcribes the precursor of mRNA, known as heterogeneous nuclear RNA or hnRNA. RNA polymerase III is responsible for the transcription of tRNA, 5' subunit ribosomal RNA or 5SR RNA, and small nuclear RNA or snRNA.
Yes, three types of polymerase enzyme. Okay, RNA polymerase 1, which will transcribe all sort of RNA, rRNA, ribosomal RNA. Okay, while we have to remember polymerase, RNA polymerase 2 is responsible in the formation of hnRNA, heterogeneous kind of RNA, that is mRNA, which will be formed later into homogeneous. Okay, it is hetero because of the exons and introns and capping, all that. That's why it is known hetero. So RNA polymerase 2 is transcribes hnRNA heterogeneous, while RNA polymerase 3 transcribes tRNA, that is adapted molecule or the transfer RNA, as well as the subunits of rRNA, some of it. Okay, so these are the three, and with names you have to remember, because this is 1 mark question: which RNA polymerase enzyme is needed for the transcription of which type of RNA. Okay.
The processing of hnRNA marks another major difference between prokaryotic and eukaryotic transcription. The mRNA in prokaryotes does not require any processing. However, in eukaryotes, the hnRNA is inactive. Since it has a split gene arrangement with exons and introns, and hence it has to undergo processes such as capping, splicing, and tailing. The first process is capping, which involves an unusual nucleotide called methylguanosine triphosphate being added to the 5' end of the hnRNA. Capping helps
learn the name. Okay, methylguanine trioside. The reason is this is asked, the name of the capping. This kind of things, small things, but then needed to learn for the exams for 1 mark questions. Now MCQs are going to increase.
mRNA to bind with small ribosomal subunits during protein synthesis. The next process is splicing, in which the introns are eliminated and exons are joined in a specific order. Finally, in the tailing process, about 200 to 300 adenylate residues are added to the 3' end of the hnRNA in a template-independent manner, giving rise to a poly-A tail. This process is also known as polyadenylation. After these processes, the hnRNA becomes mRNA and the transcription process ends. The mRNA can now be transported out of the nucleus and further used for the process of protein synthesis during translation. In 1962, James Watson and Francis Crick won the Nobel Prize for Medicine for deciphering the structure of DNA, the molecule that contains an organism's genetic blueprint.
Okay, so this is transcription is getting over here. Okay, anybody wants to ask anything? As I said, that they are not explaining you separately transcription in prokaryotes and eukaryotes, because the process is same, except the 3 extra steps which is happening. Okay, the 3 extra step happening only in eukaryotic type of transcription, not in prokaryotes. Okay, and the 3 steps: number 1, the first one is capping. Okay, the capping is that is, it is written, if you write, it is capital G and triple P. Okay, then after capping is done, then splicing is the cutting of the introns and coming off together of exons, while polyadenylations are nothing but the addition of many adenines, okay, in the 3' end or 3' end of the mRNA. And after this 3 process, now it comes out of the nucleus. Anybody wants to ask anything on it?
No, ma'am.
Okay, so there are
No, ma'am.
actually only one I mean, what you call it, this mRNA is formed the same way it is now. You are going not, they are not going to tell you about tRNA, rRNA and all. Okay, by the same process it is formed. Like they have told you what the difference is, only the type of enzyme. Okay, in eukaryote, the enzyme, that is polymerase RNA polymerase enzyme used for making tRNA and rRNA is different in eukaryotes, while in prokaryotes it's again the same polymerase, RNA polymerase, while only the difference comes in the type of enzymes of RNA polymerase enzyme. So they are not going to tell you how the transcription of T and rRNA happens. Okay, so we'll move forward. 5 minutes are
Yes,
left.
ma'am.
Yes, ma'am.
Through
Yes.
experiments, Watson and Crick had revealed that DNA was a double helix polynucleotide molecule, with each nucleotide consisting of the deoxyribose sugar, phosphoric acid, and nitrogenous bases adenine, guanine, cytosine, and thymine that are commonly called nucleotides. After the DNA structure was revealed, these scientists got busy finding an answer to the next question: how the nucleotides get converted into amino acids that synthesized proteins, the building blocks of life. These scientists already knew that DNA directs RNA synthesis, which in turn acts as a template in the synthesis of protein. They therefore suspected the presence of a genetic code, a set of rules in a gene that instructs a cell on how to synthesize a particular amino acid from the nucleotides of an RNA strand. The scientists now had only one aim: to decipher this genetic code. George Gamow, a Russian physicist, was the first to achieve some success in deciphering the code.
Okay, uh, genetic codes are nothing like, but then very important information regarding now, if you are getting any medicines, if you are getting any kind of vaccinations, if you are getting anything biogenetically made, is because of the discovery of this genetic codes. Okay, after we are going to learn about genetic codes. Like there's a chart in your book of genetic codes in which about 20 amino acids codings are given. Okay, and in that you'll be learning about the codes for each protein. Okay, and how it was, we are going to the history of discovery of genetic code, how it came into the triplet only is the coding, not the quadruplet, and all those things we are entering into. And out of this, when you look into the book, kindly learn few codons which you have to remember. As it's writes for valine, you have to learn for some of it is important, like very important. If you can learn all, it's well and good. Like stop codons, which are start codons, which are, these are again 1 mark MCQ questions, or it can be even 1 mark subjective questions. Okay, so start codons, stop codons, these are the codons. Will stop and start codons means it will help in starting and stopping of the protein synthesis. Okay, so like that it you have to learn. So George has done a great work, and we are learning ahead about it, how, and we came down to the conclusion that it is a triplet which codes for each amino acid.
He initially suggested that since the mRNA strand consists of only 4 types of nucleotides that code for the 20 amino acids in the human body, they need to combine to form a particular code. Through mathematical formulae, Gamow then proved that a permutation and combination of 3 nucleotides would be sufficient to make up a specific code for each of the 20 amino acids. The logic behind Gamow's postulation was easy to see. A single nucleotide, for instance, would code only 4 amino acids. Likewise, the combination and permutation of 2 nucleotides would result in codes for only 16 amino acids. However, the combination and permutation between 3 nucleotides would code for 64 amino acids, a little more than required. But nonetheless, Gamow stuck to his postulation. Though scientists were now aware that a combination of 3 nucleotides was all that was required to make a particular code, they had another question. Which combination of nucleotides coded which amino acid? The answer to these questions came from research scientists Marshall Nirenberg and Heinrich Mattei. They created a free cell system by grinding E. coli bacteria using a mortar and pestle to obtain E. coli extracts. The E. coli was then added to a test tube containing everything needed for protein synthesis, including ribosomes and amino acids, to name a few. However, the test tube lacked an mRNA, without which protein synthesis was not possible. The scientists then added artificially synthesized mRNA consisting of only one base, uracil, in the free cell system. The E. coli extract in the free cell system was now able to produce a protein chain made of one repeating amino acid, phenylalanine, with the help of the Severo-Ochoa enzyme called polynucleotide phosphorylase that was present in the extract. This suggests
this was the experiment. After this, many experiments they conducted like this to give us that 20 codes for the amino acids. Okay.